This presentation will give you some idea of some of the benefits which can result from using silicon nutrient in strawberry fertigation. Our work shows how the use of a bioavailable silicon nutrient can increase the resilience of strawberries to both pests and diseases, as well as giving many other benefits. You expect optimum nutrition to give you healthy plants, but strawberry powdery mildew is still a major problem, and growers are spraying with fungicide every 5 to 14 days, depending on what program they're using. However, growers do frequently spray potassium bicarbonate in a wetter, often a silicon wetter, in order to control strawberry powdery mildew, and some organic growers only use potassium bicarbonate in a silicon wetter in order to control the disease throughout the season. Our investigation started in 2008 when a grower asked us why you got better control if you were putting your potassium bicarbonate on in a silicon wetter. All plants actually need some silicon, particularly in the leaf hairs. Silicon itself is the second most abundant element on the planet, but most of it is not available to plants. It needs to be bioavailable, salicylic acid, to be taken up and utilized by plants. All plants actually have silicon transporter genes which function in the roots to bring the silicon into the plant. A lot of research has been done with monocotyledons like rice and wheat and so on, but strawberries have not had so much research done on them. The effects of silicon on the plant from the literature include increasing cuticle thickness, increase in plant hairs on the leaf surface, thicker cell walls, etc., and physiological effects including increased resilience to biotic and abiotic stress. Monocotyledons are actually classed as silicon accumulators, but strawberries are classed as silicon non-accumulators, which is probably why there hasn't been so much research on them. There's a big body of research on silicon on other plants, and most research has actually used potassium silicate. However, this does have some disadvantages, including the silicon being deposited out in the fertigation tube, so we have not used potassium silicate, certainly not on farm. Silicon is not classed as an essential element, and therefore it's not included in most plant growth media. In the trials reported here, we have been using a formulation that gives you bioavailable silicon. The formulation breaks down to salicylic acid, which is then taken up readily by the plant roots and transported through the plant via the xylem, and then the silicon is deposited in the leaf cells and alters the wax structure. And if you look at the following slides, you will see some of that. This slide shows you an early trial. I think we've now done 6 or 7 years worth of trials, but it shows an early trial of the effect of silicon, bioavailable silicon, put in the fertigation tubes on a commercial farm and conducted under totally commercial conditions. And what you can see from this trial is that the blue line is the totally uninterrupted, uncontrolled epidemic, something I hope that you never have. Whereas the red line shows you what happens if you are just putting silicon in the fertigation tubes and not using any fungicides. And if you look in the middle of the graph, you can see that the silicon alone in the fertigation tubes stops the start of the epidemic by several days. Silicon given once or twice a week in the fertigation system or as a weekly spray definitely increases the resilience to strawberry powdery mildew as seen in the previous slide. And it also retards the start of the epidemic by, in our various trials, 8 to 12 days. And the results have been obtained for 7 consecutive years and are statistically significant. There is a seasonal variation in the disease pressure, but the resilience to strawberry powdery mildew is seen in some form, whatever the disease pressure. These are scanning electron micrographs which were taken as a result of a glasshouse experiment. The top 2 pictures show you a 0.25% silicon spray application, and you can see that the wax surface is fairly, has got ridges all over it, but they're not very close together. If you look at the middle pictures, which was a 0.1% root application, you can see that the ridges that are appearing in the wax are far more dense, and if you look carefully you can see little lumps of silicon known as phytoliths. And the bottom slide is the control with no silicon application, and you can see that the ridges on the wax surface are actually a fairly wide network. This is another scanning electron microscope picture, and on the left hand side you can see what happens without silicon, where the cuticle is fairly narrow, the measurement is there, and also you can see some of the leaf hairs, and you can't see many projections from the wax. If, however, you look at the right hand side, you can see quite clearly that the cuticle and wax is much thicker, but more noticeably are the pinnacles that are on the surface, which are in fact encrusted silicon, and this was given through the roots, and they are forming what are called phytoliths on the surface, which make the surface, if you like, much more scratchy. Here we are using a molecular tracker dye which enables every particle of silicon in the plant that you're looking at to fluoresce. And so on the left we have the example of a cross section of a strawberry leaf without any silicon being given, and you can see that there is, relatively speaking, very little fluorescence. But on the right you can actually see that there is a lot of fluorescence in the leaves when silicon has been given through the roots, and you can see it in the upper and lower cuticle and in the palisade cells. You can see it in the stomata on the far right, and actually what is probably a leaf vein in the left. So plenty of evidence that silicon is transported through the plant and then deposited. This gives you some idea of how the silicon travels through the plant. On the left hand side you've got the without silicon, and this is a cross section of the petiole, but on the right you've got a cross section of the petiole where silicon was given through the roots, and you can see quite clearly the fluorescence not only in the cuticle but also very much in the xylem vessels in the center of the leaf petiole. Now here you have some cross sections of the roots using the same fluorescent dye, and on the left you will see it's a bit of green, so they are getting some bioavailable silicon, but where you've got the silicon application through the roots you can see really clearly that there is silicon deposited in the xylem in the center of the root. Now this was perhaps our most surprising finding by Dr. Beaulouis, who showed that if you had silicon through the roots of strawberry plants, actually in the field in a commercial tunnel, the infestation by two-spotted spider mites was greatly reduced. There was a peculiar effect with fungicides, which were to do with using a biological control of the two-spotted spider mites. But the bottom line is that whenever you used silicon through the roots, you got lower numbers of two-spotted spider mites. This slightly more, if you like, scientific slide shows you that these results happened 2 years running, and that there was always a statistical difference in the number of two-spotted spider mites whenever you used silicon, and it happened in both seasons, and the evidence is that it would always happen. How does silicon increase the resilience to strawberry powdery mildew and two-spotted spider mite? Well, the first line of defense in any plant is the outer surfaces, the wax, the cuticle, the epidermal cells, and the cell walls. And so our hypothesis is that because silicon gives thicker cuticle, thicker cell walls, because the silicon is deposited so clearly in the cuticle and in the cell walls of the palisade layer, that is what is increasing the resilience. And also the phytoliths on the surface inhibit the formation of the fungal aprosoria, but also inhibit the feeding of the two-spotted spider mite, which feeds by scraping on the surface. So the use of a bioavailable silicon nutrient throughout the season enhances the passive defense pathway, and so reduces susceptibility to strawberry powdery mildew and two-spotted spider mites, thereby increasing the resilience of the plant to these pests and diseases. We have found quite a lot of additional benefits of silicon, including the increased number of leaves, definitely an increased biomass, an increase in the number of runners, and flowering 7 days earlier when there's silicon present in the hydroponics, definitely increased chlorophyll in the leaves, and in the glass house an increase in bricks. We have tried to work out whether silicon in any way could be considered essential for strawberry growth. What we have shown is that the biomass of plants given silicon is greatly increased. It's as if all the plants that one's been looking at without silicon actually have been held back. They're all much smaller. The converse is also interesting. We have tried to look for toxicity, and at really quite enhanced levels, well above what we would recommend for fertigation, there have been absolutely no toxic effects at all. We've also shown that at the concentrations we are recommending, there's no change either to the pH or the EC of the fertigation. So, in summary, we believe that bioavailable silicon, when it's delivered regularly throughout the season as a nutrient, increases the resilience to pests and diseases by enhancing the physical defense pathway of strawberry plants, and that bioavailable silicon is often missing from most fertigation plans, and we also have found other benefits as well, including increased chlorophyll levels. Thank you for listening, and thank you very much to all our co-workers, without which this work could not have been done. Goodbye.